Thanks, Mark. Thanks for the opportunity to present today. So I really want to explain just for a minute why Rivanna really stands apart in the world of retina today. We believe we're one of the few companies in the world that are taking a 6-month biodegradable implant that can be delivered in the clinic, delivering biologics and also large molecules as well. So we're looking to solve the number one unmet need today in retina, and as we said, is the reduction in the frequency of injections. We believe we now have a breakthrough in biologic delivery, which has never really been accomplished before, again, to be directed into the clinic. And that's because we've developed a photocross-linked biodegradable hydrogel implant. So we use light instead of heat to develop our implants, and that's really given us a pathway forward to develop biologics. Other platforms that can PLGA do have acidic breakdown, so that's why PLGA platforms cannot deliver biologics themselves. A lot of strategic momentum behind the company at the moment. We have an innovative pipeline. We're looking at 6 months to change the release of aflibercept. We have a second-generation bispecific asset we're looking at. We have multiple strategic collaborations. We have validated preclinical results. We've put together a really quite formidable team going back to Vitrasert days, world-class advisory board. We've raised about 18 million to date, and we're now just initiating a 40 million Series B raise. So as I mentioned in the panel, we have two core platforms. One is a preformed implant procedure, photocross-linked, before it goes into the eye with ultraviolet light, which is iLeaf and iLeaf SD. And then a second in situ implant, Oculeaf, photocross-linked in situ with visible light, that can deliver biologics for between 6 months, in fact, up to 12 months with this particular product technology. The retina market today is projected to go to 22 billion by 2029. So Rivanna's currently have presence in all three segments, both anti-VEGF, bispecific, and we've also worked with anti-complement drugs. We're really embarking upon multiple de-risking programs at the moment, really with the two internal assets, and again, working with external partners, whereby we use their therapeutics to look to deliver into our technologies. We've worked actually today, we've now worked on 19 different therapeutics, so we've got an extensive pedigree of experience here. We've worked with 9 biologics, or large molecule drugs, and then small molecule drugs as well. So we've built up an enormous amount of expertise ever since Rivanna was founded about 7 years ago. We control biodegradation in a really clever way. This is some studies, histodata from a non-human primate study that we did. So as I mentioned in the panel, it's really, really important you don't show inflammation, because that really kills the technology. So we've shown some excellent tolerability results, both in this NHP study that we did and also other animal studies as well. Perhaps quite a rare occasion, we've now even shown zero-order release of a bispecific. It's very rare you'll be able to see this, to get zero-order kinetics, and this is with a bispecific product. So this is part of the capabilities of the technology that we've developed. We have a great set of investors on board. We've raised about 18 million with Visionary Ventures, Excite Ventures, and Focus Capital from the US. We have 3 main UK investors, Cubis, Techstars, Clarendon, and we also have a corporate strategic pharma investor as well. The 40 million breakdown is really taking the 6-month aflibercept into the clinic with a 24-patient study. But we've got so many other opportunities within Rivanna, both in the Oculeaf with their second asset, that we really think that will drive major partnerships going forward. The team is really steeped in ocular drug delivery, going back to the Vitrasert days that I mentioned. Our CEO is here today, Patrick Ran, who's been involved in ocular drug delivery and photochemistry for many years. We have a world-class advisory board, some of the faces you will recognize. Interestingly, Anne Doherty, he was 40 years at Genentech in ocular drug delivery, just retired from Genentech and came straight into Rivanna as an advisor. So we have a fantastic group of advisors that really help us, both investors and retina specialists, that really help guide our pathway forward. So really to summarize, it's all about now CEOs, while they're trying to raise the money, it's all about implementing the vision of the company. And that is really a clinical breakthrough by delivering sustained-release biologics in the clinic. We think this will have major industry impact as we go forward. We're accelerating our biologic sustained release development, both with AI and with machine learning tools. And ultimately, our strategic vision is to create a continuum of biologic and other ocular drug delivery products with our iLeaf SD, and our Oculeaf technology. So with that, thank you very much.
Any questions? Well, Michael, I'll throw one out. I heard or read that the FDA may be considering dropping animal trials for biologics, immunotherapy. I'm just curious if that could potentially apply to your development of biologics, compounded with polymers. I don't know the clarity of what they're throwing out. It's sort of a tumultuous time with the FDA, as we'll hear more about later in the day. But if indeed it's easier to develop without animal models, wouldn't this speed up your pathway, or is this just a pipe dream?
Yeah, absolutely. I think if you could, we actually welcomed it in many ways, that if you can avoid animal studies, particularly NHP studies, clearly, that would be a major bonus. It's not very clear exactly as to what exactly lies ahead, but we've already looked at our use of proceeds that would take us into the clinic, and we believe that there could be a faster track method of doing that if these FDA proposals actually go ahead, and it could be a substantial cost reduction as well. So we're kind of in no man's land at the moment a little bit, because we don't know exactly how this is going to work out. But if it truly materializes in the way that they have projected, we think that could be extremely beneficial to Ravana.
Yeah. Michael, question for you. I mean, obviously, to inject a already cross-linked product, that I understand, but the light cross-link, when you're doing it in the eye, can you comment? Are you doing that in the eye? Did I get that right? You inject it and you put the light in. But can you talk a little bit about what that is about? How long is the light? What kind of light? How do you cross-link it in the eye?
So the first technology is an implant. It could be 5 millimeters by 0.5 millimeter or less. So that's photocross-linked with ultraviolet light, but there's no light goes into the eye. So it's a ready-made, completely preformed, and injected straight into the eye, let's say with a 25 gauge needle. The second technology is, it's really a liquid, it's a gel. So same makeup, same chemistry as iLeaf, but injected as a liquid. So what we do is we have developed a proprietary applicator working with Cambridge Design in the UK that allows us to both inject the drug, but also the ability to photocross-link it in situ at the same time. So the gel goes into an aqueous environment like the vitreous. It forms the depot, forms the implant. We then photocross-link it, but in this case with visible light. So there's no UV light goes into the eye. So we've worked, Patrick, our CEO, is actually a photochemist by trade. So we've got this enormous amount of expertise that's allowed us to look at visible light. But the whole idea here is you would then photocross-link that in really something like 10 seconds or less. So it doesn't really take away from the daily, intravitreal requirement that you're seeing retina physicians have today. So it's a very novel approach, never really been done before. But even for the size of a 20 microliter bleb, we can get 10, 15, or 20 milligrams of drugs. So for a tiny amount of gel, you get a substantial volume of drug into the eye. So this would allow us to show that we could deliver biologics way beyond 6 months, perhaps up to 12 months in the eye with our second technology. And they're really divided by iLeaf, which is really for therapeutics that require less than 2 or equal to 2 micrograms a day. Oculeaf would be for greater than 2 micrograms a day for therapeutics. So with that wide spectrum of therapeutics, it gives us the opportunity to work with many different molecules. We believe the future is sustained release, and that's our focus.
Great, Michael. Thank you so much.